Good morning, everybody, and welcome to today's webinar that Avantor are running jointly with Tosoh Bioscience on the analysis of biotherapeutics by HPLC. So we're really looking at protein characteristics and how they are analysed. So before we begin, there's a few housekeeping things that I would like to raise. So we will send you the recording following the session. Please feel free to submit your questions at any time in the questions section on the GoToWebinar dashboard. We'll do a Q&A session at the end with Elena. We will send you a certificate of attendance following the session. And if you have any questions after the event or any additional feedback after the event, then please contact us at webinar@avantorsciences.com. So with that, I'd like to introduce our speaker. So Elena Kumm studied chemistry before she received her PhD in biochemistry with a focus on proteins in human platelets. Afterwards, she worked as a postdoc in platelet research, protein purification and plasma proteomics at the University Medical Center in Mainz. She joined Tosoh Bioscience in 2022 as application specialist, where she investigates new applications for instruments, process resins, HPLC columns, and gives trainings and technical support to customers. So with that, I'd like to pass over to Elena, and thank you in advance for this presentation.
Yeah, thank you, Cassandra, for this warm introduction. And welcome everyone for today's webinar. I will switch off my camera during the webinar, so we don't have maybe data problems or something, but I will switch it on again for the Q&A, so you can see me again. Okay, so I hope everybody's fine. You're interested and up there. So I will give you a broad overview today on how we can analyze protein characteristics of our biotherapeutics. So first, as an introduction, many of you, I think, already know that biotherapeutic samples are not a homogenic solution. They often, after production, you have biotherapeutic samples that might contain different variants of your protein of interest, for example. So this might be fragments, it might be aggregates, or incorrect formed disulfide bonds, methionine that are oxidized, isomers or N-terminal cyclizations. But it might also be that your target molecules have different glycosylation patterns. And these might be naturally occurring patterns, but some of these, let's call it impurities or modifications, could be also caused by improper storing of your sample. And the storing is leading to stress for your molecules, of course, and then it might also be that these samples are degraded. And also when your sample is a bit older already, also these modifications might occur. There are, of course, also cases where you might want to modify your target molecule by purpose, like pegylation, for example, or you want to have some isotopes. But even there, your solution will not be homogeneous at all. And the problem with impurities or protein variants is that they might behave different than your real protein of interest. So, for example, they might have different immunogenic effects on the body of a person if you give that to a person as a drug, or they have other binding kinetics of specific protein-protein interactions. They also might have different pharmacokinetics or different serum half-life. So long story short, they all have an impact on the efficiency and the security of the biotherapeutic solution. And therefore, it's really important that your sample is analyzed and maybe also, if it's necessary, separated before you want to use it. So there are different, of course, chromatographic modes to unravel these different protein characteristics. And I here summed up all most commonly used modes to analyze your sample. So this is, for example, maybe your sample contains something like aggregates or fragments, then you're able to separate your sample and the different impurities by their size. And this you do with size exclusion chromatography, also called SEC or gel filtration. Then, of course, it could also be that your molecule has different charges, different molecules with different charges in your sample. This could be caused, for example, by methionine oxidation, cellulation, or deamidation. In this case, you use ion exchange chromatography, also shortly called IEX. Then, of course, you could also have different hydrophilicities of your molecules that are maybe caused by glycosylation patterns. And therefore, you use the hydrophilic interaction liquid chromatography, also called HILIC. Then it might also be that you want to investigate the hydrophobicity of your sample. That might be caused also by deamidation, isomerization, aggregation, or fragment. There you can use hydrophobic interaction chromatography, also called HIC, or reverse phase chromatography. But today we're focusing on the HIC process. And then there's, of course, also one mode that I think is quite common, and I really like it. It's the affinity chromatography. It's also really important. This you use often to determine the titer of your sample. And yeah, you have protein A or L, or you can even go through the FC receptor. And there it works that protein A and L, or also this receptor, have an affinity to the FC part of the antibody, if your sample is an antibody, of course. And yeah, then it can just interact with this. But I will go into details for all of these methods in the next slides. So I'm starting with the mode that is very popular for protein analysis, but even though is not very specific, the size exclusion chromatography. So it's a very gentle separation mode, as you can use physiological conditions with a pH between 6.5 and 7.5, and also medium salt concentrations in buffers. So therefore, it's possible to analyze native proteins and also to preserve their biological activity. SEC is a non-absorbing technique, but however, the peak capacity of SEC is low compared to the absorbing techniques that are used otherwise. The separation, and that's really important, is highly depending on the column length, the particle size, but also the flow rate. So also be aware of this. Here I show you the separation principle of SEC. So ideally, you don't have any interaction of your molecules with the stationary phase of your column, of your SEC column. So then you're able to separate your sample by size, as the name says. And therefore, we have a porous stationary phase. So here is your sample, all these colored dots, and you see you have different sized molecules within your sample, and then you add them. And the larger molecules, like the red dots here, are not able to enter the pores of your stationary phase. So they will flow fastest through your column and will be eluted first. And then, of course, depending on size, the smallest molecules, but also ions that are derived from your buffer, for example, will enter the pores and therefore will elute later because it needs them much longer to cross your column. It's like these blue dots here. An example, so in which case would it make sense to analyze my sample with SEC? In this case, if you have a monoclonal antibody, for example, this is your desired product, your antibody. But of course, it might be that you have some impurities like aggregates or fragments that you don't want to have, especially as fragments are non-functional, immunogenic, and then also have another half-life within the body than your antibody. And aggregates also some of the same. They're less efficient and they're also immunogenic, so they shouldn't be in your sample in the end. So you take all your sample, and in this case, we're using an SEC column, TSK JLG 3000 SWXL. And we're using as buffers the potassium phosphate buffer and potassium chloride. And we are now, if you see here the diagram in the end, we are able to determine the impurities and even their amount in percent within the sample. So you see that you have your main peak of your product, of course, your monomer, but then you have before this is another small peak that appears. And this is like, as it elutes first, it must be bigger than, like larger than your molecular, higher molecular weight. So potentially these are aggregates. So some higher molecular weight, whatever sample, this is what HMW means, higher molecular weight. with, in this case, let's say 4%. And then we have a separate, another peak, another small one that elutes later than your main sample. So it must be some smaller impurities, maybe some Fab fragments or something else that is smaller from your antibody. And these are the lower molecular weight molecules. I mean, you see that we even can determine the percentage, but of course, this also has limits. It, of course, also depends on how much impurities you're still willing to accept in your sample. I mean, if you say, I really just need 0.1% or something, this might reach the limit of the column. But I mean, then you have also other methods to determine these impurities. But if you say, okay, this is fine, I don't even need to separate now, it's okay with these low amount of impurities, I can work with that. You even just did your analysis and then you can work with your sample, which is really good. You could, of course, also use SEC if you have different molecules in your sample and you are sure that you have them, maybe not an antibody, but something else. But you're not really sure what proteins these are or what sizes they might have. So you just run an SEC, you get different peaks. And if you're really interested in the molecular weight, you could, for example, also use light scattering detector afterwards to determine the mass of your different peaks, or also mass spectrometry, of course. The next mode that I want to speak about is the ion exchange chromatography. So it might be that your impurities or modifications have the same size, so you can't use SEC, but they maybe have different charges. So in this case, we have also then maybe different separation patterns of the differently charged molecules. And we are then able to distinguish between the different variants just for the chromatogram when we look at that. So the principle of ion exchange chromatography is that charged molecules are binding to the stationary phase. That stationary phase should have an opposite charge, of course, and this should be ideally at no or low salt conditions. So there are rarely or nearly no salt ions present within the solution when you're starting, or when you bind your sample also. And these molecules that interact with the stationary phase are then eluted with the salt gradient. So therefore, you of course you're increasing the amount of salt that you're adding, and the charged molecules on the stationary phase are then exchanged by salt ions. So the elution pattern that appears is, of course, according to the charge of the molecules. So low charged molecules, it will elute first and are then followed by higher charged molecules. You could, of course, also, if you say, I don't want to have so much salt in my sample, you could run, if it's possible, a pH gradient instead of a salt gradient. And this then, of course, is not like that the salt ions are then replacing your molecules, but that the pH has an impact on the charge of the analytes. And this changes also their charge. Yeah, sorry. And then, of course, you don't have this competitive binding of salt, but you still elute within, depending on what pH you're going, increasing or decreasing pH, you're eluting your molecules again. Yeah, the protein charge is, of course, important. You should think about what charge you might have and therefore, what separation mechanism you want to use. I mean, if you have anions, you will separate them with anion exchange chromatography. This is often used for example, for oligonucleotides, plasmids, mRNA, or viruses. And then on the other hand, of course, if you have cations, you will use cation exchange chromatography. And this is often the case for many proteins. You see here also as an example, the charge of the molecule is depending on its isoelectric point. So this means that you should always think about what pH you want to use, and you should think about the isoelectric point of your protein. Let's say you have lysozyme with a quite high isoelectric point of 11.2, and you run your chromatography with a pH of 7. So your lysozyme will be positively charged at this pH. So you need to be aware of that, what conditions you then need to run, what you need to use, because otherwise you don't get a separation. Of course, you can't isolate your sample. On the other hand, for example, BSA has a really low isoelectric point of 4.6. So at pH 7, it would be negatively charged. So you would need to use another mode, or you need to use another buffer, or whatever, another pH. So just think about before you start. Then, of course, you also have charge variants of your protein. And these can be due to modifications of your protein, like, for example, oxidation, deamidation, or post-translational modifications like phosphorylation, for example. And these modifications then, of course, are influencing the binding kinetics of your sample. They influence the protein half-life and also its activity, of course. So therefore, it's really important to determine your modifications, to be aware of them. And many of them might be caused by aging. You see here on the right side as an example what that means, like modifications. So you have this cyclization of glutamine that leads to puroglutamate, and this leads to a less positive protein. And the same also if you have deamidation of, for example, here asparagine to aspartic acid, it will be also a less positive protein. This deamidation often occurs at higher temperatures or at a pH above 10. So if you're using normal, like, physiological conditions, this shouldn't happen. But I mean, always think about how to store your sample, how you're treating your sample in between, and what might happen because of this treatment. Yeah, and somehow with ion exchange chromatography, you're looking at the percentage of the basic and acidic charge variance of your protein that you might have, if you look at it. Here's an example for cation exchange chromatography. So we have a monoclonal antibody that has a charge heterogeneity. So we have different charges within our antibody. You also see that we have acidic variants and basic variants. And we have two different cation exchange chromatography columns. In this case, it's two TSK gel columns, the CM stat and the SP stat. And these both, I mean, they are both cation exchange columns, but they have on their surface, they do have cation exchange groups, and they can separate with a very high resolution differently charged isomers of one monoclonal antibody from the main project when we are running a salt gradient. As you also see in gray, this is the salt gradient, or in the description. So then we have in red, we see the CM stat column, and in blue, we see this SP stat column. And I mean, if you look at these two, if you look at the two chromatograms, you already see that they have different selectivity for these different variants. So they are differently strong cation exchangers. And the blue line, the SP stat, is the stronger cation exchanger in this case. And obviously, this one now is leading to better separation of the variants. So for this case, you would say, okay, I use the SP stat because there I can really detect my different variants. But often it's hard to say in advance which of these two columns would be the better choice for your specific sample. So it makes sense often to just do test runs if you're not sure. And often people use weaker cation exchanges because there they have more options to influence the selectivity of the column. But it doesn't mean that this is the rule of what you should always go for. The next mode is the hydrophobic interaction chromatography, also called HIC. So your target molecules could be not only influenced by different charges, but also by different hydrophobicities due to the modifications and impurities. So if you want to analyze the hydrophobicity, you use HIC, as I said. So which HIC is a weak hydrophobic, it has a weak hydrophobic stationary phase. So this. And this one can then interact with hydrophobic regions of molecules. So first, the columns are equilibrated with a high salt buffer, and therefore we have a low solvation of the molecules and the hydrophobic patches are accessible. So the hydrophobic patches then of the protein and the ligands on the surface are interacting, and because of this, water molecules are released and can gain a higher degree of freedom. So if you want then to elute your sample again, the salt concentration is decreased and the solvation of the molecules gets higher with this, and then they are eluted in order of increasing hydrophobicity. So the more hydrophobic region it has, the later it will elute. And of course, protein have different hydrophobicities, not all of them are the same. So the good thing about HIC is that we can work at non-denaturing conditions. And yeah, I mean, you see already here on the picture, the red parts are the hydrophobic regions of proteins. So you see that there are different amounts of hydrophobic patches on the surface of proteins. For example, an antibody has, always depending on the region, on the Fc part, a very high amount of hydrophobic patches. And these hydrophobicities also can be influenced by different modifications of proteins that can occur also during aging, for example, or maybe other treatments. So the modifications that are impacting hydrophobicity are, for example, deamidation, as you see here again, or the methionine oxidation. This is decreasing hydrophobicity. But on the other hand, you have like aggregation or fragmentation, what is increasing the hydrophobicity of your sample. And then, of course, a special point is if you have antibody drug conjugates and your drug is hydrophobic, the more drug you're adding to your antibody, so the higher your hydrophobicity will be. So if you know what your drug is and how the hydrophobicity is, you could also guess already how your sample might behave in chromatography. I also have for this, I have an example for you for HIC to analyze the antibody drug conjugates. So here we use a TSK gel butyl NPR column, and you can nicely see that you're eluting your sample with an increasing amount of drug somehow. So if you have no drug coupled to your antibody, it elutes first because it's not that hydrophobic, but the more drug you have, the later it's eluting, and it's also making a really nice and clean separation peaks. So you really see what's going on and what you might have in your sample. Another example for HIC is if you have the idea of exchanging Fab arms of your antibody. So there are cases where you have like B-specific antibodies that you're matching, and then you want to end up with a multi-specific antibody, just as an example. I mean, this is of course only suitable to use HIC there if you have different hydrophobicities of your sample, because if they're all the same, you won't see any differences, of course. But you can nicely here see in the diagram that the first B-specific parental antibody is eluting first because it's not really hydrophobic. Then comes your multi-specific product, and then in the end, the more hydrophobic B-specific parental antibody 2 is eluting quite much later. So this is a very helpful method if you want to check the recombination efficiency of your reaction. And it's, yeah, it looks quite easy, right? Doesn't mean that it's always like that, but it's really good. Another example again for HIC is if you want to look for the oxidization state of your monoclonal antibody. So here we use a TSK gel butyl NPR column, and the monoclonal antibody has been first digested with papain. So we get Fc parts and Fab fragments, so we don't have a total antibody anymore. And then it has been treated with tertiary butyl hydroperoxide, which is also called TBHP, and it oxidizes methionine residues on the antibody. This is now, of course, artificially made, but it could also happen just by storing your sample or treating it wrong. So it's how you could see if something happened to your sample. So in general, the more TBHP we are adding, the higher the amount of methionine residues that are oxidized. So the blue line shows the chromatogram where no TBHP has been added. So you have two peaks. You have your Fab fragment peak that elutes first, and later you get your Fc part. As I told you, it has a higher hydrophobicity. And then when you're adding 0.1% BHP, so the green line, you see that the second peak at 16 minutes is splitting into three peaks. I mean, the third is a bit small, but yeah. The highest peak is still non-oxidized methionine residues, so you don't have any reaction. But the second peak is Fc part with one oxidized methionine residue, and the smallest one is then the Fc part with two oxidized methionines. So the red curve here in between, you see you just took less TBHP, but you already see that there are some small peaks appearing just in parts. I mean, it's quite tricky to see, but you already have an idea that something will happen here. Another mode that I want to speak about is hydrophilic interaction chromatography. I already told you it separates molecules that are differently hydrophilic. This could be, for example, different glycan patterns that are released from proteins or antibodies. So in HPLC, we have a polar stationary phase. This is here in gray. And the mobile phase is acetonitrile with a low amount of water. This is really mandatory. And due to this, then a thin water layer is formed at the stationary phase here. And yeah, your hydrophilic analytes will then separate within this water-rich layer. And then they can be eluted if you're increasing the amount of water within the mobile phase. So the most polar compounds will elute the latest with the highest amount of water. The beneficial thing about HPLC is that you can directly use it with mass spectrometry, for example, because you already have an organic buffer that is, for example, acetonitrile. That's really good, I have to say. Yeah, different glycan patterns are differently hydrophilic. So, of course, they impact the protein structure and the function of your protein. But if you look here on the right side, you see that depending on your glycans, they have different influences. So it might be for fucosylation, for example, it reduces the immune response, whereas the galactosylation and the B section are increasing the response. And there's also cellulation, on the other hand, that is increasing the antibody half-life and the anti-inflammatory effect, as well. So it makes sense to analyze your glycosylation patterns if you think that you might have different ones before you're using your sample. This analysis is a very important and a multi-step process. So first, the glycans are enzymatically released from the protein. Then they are fluorescence labeled because glycans don't have a real big UV signal, so it's quite low. So you need to have another signal that you can measure. And this is then followed by a cleanup step, and afterwards they can be separated by HPLC, and then the fluorescence detection is done, or you do mass spectrometry afterwards. Here you see a hydrophilic interaction chromatography with different glycosylation patterns. This can look like this. We use the TSK gel amide 80 column. And we use as a sample, we use the 2-amino benzamide labeled human IgG and linked glycan library. You see the overview here. And you see you can detect the signals with fluorescence quite nicely separately. So you really get an idea of what glycans might be in your sample, what not. If you've run a comparable sample like a standard with it also. The last mode that I'm presenting, but very important one, is the affinity chromatography. So molecules are captured very specific with affinity chromatography, as you have ligands on the surface that interact with your protein of interest. So it's a really specific interaction. And the target molecule can bind at physiological pH, which is really good. And then you are eluting with a low pH or decreasing pH gradient, depending on what you would like to see or what you want to have. And your eluate is then containing only the target molecule, so you have a very clean sample. You could, if you're interested, further analyze this, or you can also determine the concentration of your sample with this method. It's quite well, especially for antibodies, because what you see is we have as a ligand, for example, protein A bound on the column, and the interaction site that is protein A interacting with on your antibody is the Fc part of the antibody, and it's often used to determine the titer of your sample. But another ligand that is maybe not that known, I would say, is the Fc gamma 3A receptor. We have a column that uses this one. Fc gamma 3A is interacting specifically with an Fc part of an antibody depending on the glycosylation pattern of this antibody. So often this Fc gamma 3A column is used for upstream processing development or cell clone screening and functional analysis. So the higher the affinity, the higher the retention time of your sample. Somehow makes sense. And I thought, as maybe not all of you know this Fc gamma 3A receptor column, I would just show you some slides in the end about this, because a few years ago I also didn't know that this exists. So we have in general the Fc receptor, the Fc gamma 3A, which is a receptor on immune cells normally, and it was made to bind antibodies. Like it was not made by an antibody, it sounds stupid, but okay. Antibodies will bind on your target cell, whatever it is, maybe a cancer cell, for example, or some bacteria. And then the Fc gamma 3A receptor on, for example, natural killer cells is binding on this antibody. So the binding of IgG to Fc gamma 3A is inducing the release of cytotoxins, and it also induces the cell death of the target cell. And this process is called the ADCC, what means antibody-dependent cell-mediated cytotoxicity. It depends, and this is the interesting thing, it depends on how the antibody is glycosylated. And we now we mimic this process on our HPLC column. So we imitate this Fc gamma 3A receptor of the NK cells on the stationary phase of the HPLC column. And then you can inject your antibody sample. Your antibody will bind to the receptor weaker or stronger, depending on the glycosylation pattern. And then when you're eluting, you use also a pH gradient. So with a higher pH, lower affinity antibodies will elute. And the higher the pH, the lower the pH, the higher the affinity of your antibody to the Fc receptor. So it's suitable to determine Fc receptor affinity. Also, and this is often interesting for biotherapeutics, also the ADCC activity and the glycosylation estimation can also be done with this. And you can also use it with supernatant or purified samples. So it doesn't matter what, you don't have to clean up your sample before, you can just run it and see what happens and get an idea of what's in your sample. It's a really nice method. So, okay, with this, I reached the end of my talk. I hope you gained now a good overview about the different chromatographic modi, we can use for analyzing our samples, our biotherapeutic samples. I thank you for your attention, and I really look forward now to the Q&A part. In case your questions might not be answered, or there's something else you would like to discuss with me or somebody else from Tosoh, just don't hesitate and contact us directly, like write me an email or call me. And we can discuss maybe problems you would like not to discuss here, or maybe you get them later and you would like to ask us something. So thank you for your attention.
Thank you, Elena, for that very informative presentation. Now, just a reminder for anyone who does have any questions, please put them in the question section of the dashboard. In the meantime, there are some initial ones. So, are there general recommendations according to pH for the buffer and the PI of the protein as a starting point for method development?
Yeah, it's a tricky question. I mean, it's an important question, but the best thing to start officially, if you never ever tried your sample, to do any chromatography things before with your sample, is to start at a pH where the protein doesn't have any charge. So the pH that is equivalent to your isoelectric point of the protein, because then, of course, you're only separating by hydrophobicity. And you could then afterwards still see, I change my pH a bit, in what direction, what happens, but I mean, only change one setting, just change the pH and not also the flow rate in parallel, for example. What you also could do, you start with the lowest amount of salt that is possible, and then you can still increase if your separation doesn't look good. You have like the common somehow, but you can't say that really generally, but often it's for smaller proteins better if you have a higher salt concentration compared to larger proteins. Small, I mean like 20 kilodalton, and larger, I mean like antibodies, for example, with 150 kilodalton. I would say like take for larger proteins maybe 1 molar of ammonium sulfate, for example, and for smaller proteins maybe 3 molar of ammonium sulfate. But it always depends on the sample, of course, and yeah, just start with a neutral pH for the isoelectric point and then see what happens. I hope this answered the question.
That's perfect. We've got one here. Do you also have IMAC columns. So I guess that's immobilized metal affinity chromatography columns.
That's a good question, because when I was at university still, I was working with these. Unfortunately, we don't have them, so you can't do that with our columns. The only thing, I don't know why the person might have asked, but I would guess maybe because you, for example, have a tag on your protein or something and you would like to purify it or analyze it at least. The only thing we have is tresyl columns. This means that you could bind whatever ligand you would like to have on the surface of your column, and then you can like purify or analyze your sample with this ligand you bound on there. Let's say you're what doesn't make sense, but on your own you're binding protein A there, and then you use your antibody, or you have a specific protein interaction that you would like to use. This is the only thing we have, but not IMAC, unfortunately.
I hope that answers the question. So for ion
Didn't completely
exchange,
answer the,
sorry.
yeah, sorry.
Yeah. For ion exchange, how do you determine whether you need a strong or a weak cation exchange column?
That's the tricky point, right? I tried also to explain before, but yeah, it's hard to say that. I mean, I know that often, depending on where you're working and what you're doing, you don't want to buy a lot of different columns, but often it's really hard to say before if you're the stronger or the weaker exchanger would work better. So the best thing is really to test both and then see how it looks and what looks better. I mean, often, the point is, if you have a strong cation exchanger, you have the same ionization of the column, like of the material, at different pHs. So it's like easier to work with that, but it's less versatile. So in case you take a weak cation exchanger, it's of course ionized at different pH values differently, but it's more sensitive, and of course you can influence it much better. So often customers decide for a weaker cation exchanger, but it doesn't mean that it's for the protein of interest really the best choice. So as I said, the best thing is really to try a bit of both variants. I hope this answered the question.
Perfect. Just looking at other questions. I guess this is probably very relevant to a lot of people, as I know we're seeing a lot more people using MS across the board. So which of these methods is probably best, most compatible with MS?
It of course also depends. I mean, the really best method to use is the HPLC, because you don't need salt, and you have your for example, acetonitrile already in your mobile phase. Fc gamma 3A affinity is also working quite well with MS. In general, of course, you could also go for size exclusion chromatography. It's also used already. Or ion exchange chromatography. You just have to take care that you adjust your buffers. So you need to use some volatile salts. So for example, ammonium acetate or ammonium bicarbonate or citrate might maybe not be suitable for every molecule you're looking for. Of course, you can go for desalting steps in between, but then of course you don't have an inline chromatography and MS measurement. For ACC, it might be that you're losing some resolution then, of course, if you're using a not quite suitable buffer for the method, but better suitable for MS. But it's also something, I mean, if somebody has their specific point and a question, they can also contact us and ask if we have recommendations for that, because I mean, we're happy always to support, and I can just tell you there are people using all these things. What is not good would be HIC. This is nearly impossible to use for MS because you have such a high amount of salt that it's just not a good idea. I mean, I was working with MS and I know what salt does. It's not good. No.
Great. We've had a little flurry of questions here, so I'm trying to keep up.
Okay.
Do you have SOPs or showcase protocols available, for instance, for monoclonal antibodies to download from your site?
Yeah, we have, if you, for example, Google specific columns or even the modes, we have application notes for many cases. Of course, we can also, if you have a really like specific case, you can just contact us and we have also customer support. We can directly help you with your specific case, but often, like for example, for these SEC columns and stuff, if you just Google the column, you will find it. But if you go on the Tosoh web page, this is also what I saw before I started working here. We have all different modes explained, and then you can, of course, go for the different columns, and there you see the different application notes that we have that are in specific cases, often for monoclonal antibodies, because that's a quite known thing for customers, for us. And you can also download them all. They are free officially, or maybe you have to register, but nobody will charge you for that or something. And as I said, if you ask us in person, we can also support you and send you some information directly.
I would also just add that if you're not very confident in reaching out to Tosoh, then Avantor have crom support at avantorsciences.com, and they work closely with Tosoh as well. So, you know, can help you find the right resources through our services as well.
Exactly. So every company where you
so there's loads of options for you.
Exactly. Don't be scared. Just contact people. They always, or check pages, but then contact if you don't find information. We are all happy to help you.
Yep.
Like me.
How would you, this is quite a big question, so it's quite broad. How would you start to develop the method on an ion exchange column?
Yeah, it's I totally understand where this comes from because I know this. You just don't know what to do in the beginning. I would say look for, I mean, maybe you know what you would like to analyze, so you know your protein of interest, maybe. If you know that and you could estimate the isoelectric point, there are also ways to calculate that. You know when it's maybe negatively or positively charged. So you at least get an idea what, because you should maybe, depending on what it also is, use physiological conditions more or less. So you don't want to work in a pH of 2 maybe or something. So depending on the isoelectric point, you can already guess what, at least what pH might make sense. And then, yeah, use like standard buffers. Maybe check what I always did that earlier. I always looked what the recommendation for the columns is that I might use, like for some cation exchanges, for anion exchanges. And you can make a guess if this buffer and salt and whatever they use might be suitable for your sample and just start with that. I mean, maybe somebody else already was analyzing some sample that is comparable to yours, or there's an old paper saying something. Just try to, yeah, it's hard. It's hard. Contact us or somebody else who might have an idea so we can help you. It's really, I think, often when you're alone with your method and you want to do something, you're a bit lost and you don't know what to do in the beginning. Totally normal. And then you don't dare also to ask people from companies. But I mean, if nobody else in your whatever where you're working research group or somebody can help you, then it's really sometimes the easiest to just ask people selling these columns and say, hey, what would you recommend? You don't have to tell us all details. Just say, I have something with an isoelectric point, maybe of this and this. What would you recommend to do? Or yeah, I hope this answered it a bit. I mean, it's hard to give a general recommendation if I don't know the sample. So
That's understandable. So hopefully that's helped.
yeah, and as I said, if you
Helped
adjust
with a starting
parameters,
point.
only adjust one by test. Don't adjust three directly together, and then you don't know which had the biggest influence or what happened. So just start with something and then, yeah.
Great. So there's then one, obviously the focus has been on quite large proteins for this talk, but there is a question here on, is there a method for small, or recommendation, I guess, for small peptide purification?
Yeah, I mean, it depends how small your peptide is. If it's not too small and you have bigger samples that are having a different size, of course, you still could go for SEC, or if it's maybe hydrophilic, you can also use helic. It depends always on what peptide you have and how it looks. If it's really, really small, of course, you will even lose the hydrophilic part, and then it's tricky. I mean, then you could, of course, go for reverse phase. This is what most people do for peptides, right? But yeah, even reverse phase, you don't need to have MS behind, right? You don't have to think that this is just necessary, but yeah, it depends. If it's not too small, it's still okay with columns. Just adjust the pore size that the column also has, that it is suitable for the size of your peptide. Yeah.
Perfect. And I've got one more here. So how do you choose what SEC column is good for that application? What tends to be the cutoff range? I assume that means in terms of molecular weight.
Yeah. Somehow we always say that the pore size should be like, I think 2 to 3 times bigger than your molecule. So make sure your molecule is fitting in the pores, but not everything else. That is, I mean, it depends, of course, what molecules you're looking for. You need a good separation. So important for SEC is, as I said, the pore size, the particle size as well, depending on what you have, the column length as well, and the flow rate. So you might choose a column that is from particle size and pore size suitable for the size of your molecule, but maybe you don't get a good separation. So then you might need to adjust some parameters like flow rates, just lower it down. The lower you flow, not too low, of course, but the better it's often for the separation of the peaks. And the longer the column, also often the better. Yeah, and then also smaller particles are often better. It depends on your sample you have. Often, I mean, on homepages, if you Google for columns, you see recommendations for the sizes of molecules, or you see already with these application notes, for example, you can see, what has been done with this. Is it suitable for the size of my sample? If it's totally different, then don't do it. I mean, and then we, of course, for example, I don't know how others also have that like this. We also have columns that are more suitable for biomolecules and some are more suitable for polymers. So just be aware of what you want to purify and what is recommended for the column. Yeah. Was that the question? Somehow did I answer it, or? Yeah. I mean, it's hard to, I can't name you one column that is perfect because it depends. So
I think that's a great answer. As you say, it's always application dependent, so
yeah, I
it
mean,
can
if
be
you're really
tricky to
unsure,
give
just
much more.
contact somebody. I can always say that. It's, yeah, we are happy, or you are happy if you don't dare to go for us, but for VWR. No.
Yep. If there's no more questions, I'll give you a minute or two to add some extra ones in the chat, if in the questions box, if you want. However, in the meantime, I'd like to thank Elena for her excellent presentation and, the wide range of information she's provided in response to your questions. As she's highlighted, all at Tosoh are very much available to contact and have those discussions. We also have a great Chrome support team as well, so you can always reach out and we can kind of connect you as well to Tosoh if needed, or kind of give you the resources that you perhaps need. So with that in mind, I can't see any more questions. I will close the session. So thank you very much, Elena.
Yeah, thank you also. It was really great to be here today and to also make a live webinar again. That's like really nice. Thank you.
Great.
Bye.
Bye.